Hey everyone, now let's talk about some environmental factors that will influence your microbial growth. So, microbes, like I've said multiple times, can inhabit pretty much all environments. And depending on the type of microbe, they can be considered extremists or extremophiles. The major factors that influence microbial growth, though, are your temperature, atmosphere, pH, and the availability of water. So let's look at these a little closer. Oh, hello. There we go. So there are 5 groups of microbes associated with temperature, and each of them has an optimum temperature where growth is going to occur very rapidly. And then off on either side of that optimum, you're going to have slower growth. So we have psychrophiles that grow anywhere from -5 degrees Celsius all the way up to 15 degrees Celsius. Their optimum growth is going to be around 10 degrees Celsius. So they can grow at very cold temperatures. We have psychrotrophs that grow anywhere from 15 degrees Celsius to 30 degrees Celsius. And their optimum is about 22 degrees Celsius. Or 22 to 25 degrees Celsius. And so they're going to grow really effectively in environments that are relatively cool, all the way to about room temperature. We have mesophiles. Mesophiles grow anywhere from about 20 degrees Celsius, 25 degrees Celsius, actually about, I'd say 20 degrees Celsius to about 45 degrees Celsius. And their optimum is right around 37 degrees Celsius, which is also body temperature. Thermophiles can grow anywhere from about 45 degrees Celsius to about 70 degrees Celsius. And their optimum is going to be around 65 degrees Celsius. And then you have the hyperthermophiles that grow at extremely hot temperatures. So long as it's above 70 degrees Celsius, they typically can grow pretty relatively well. You're not going to find humans in the hyperthermophile environment, and you'll find humans living in the areas where there are psychrophiles, but not in the environment of those psychrophiles. They'll have a lot of warming agents around them. They'll be living in something that keeps them warmer with typically heat, because otherwise they could die. So the organisms that typically are around humans are those psychrotrophs, those mesophiles, and even thermophiles to some extent. So psychrotrophs are often thought of as your food spoilers. So they're going to be in the refrigerator. They're going to be the fungi that grow on foods, bacteria that grow in those foods, and they spoil the foods. We have mesophiles that are your typical pathogens. So these are the organisms that live so they are part of our normal flora, but they're also the most common pathogens. So how are they able to survive in these different environments, these different temperatures? Thermophiles have special proteins that resist denaturing. So the reason most organisms can't survive in those high temperatures is because proteins denature at a high temperature. But if they have special proteins that can survive those high temperatures, then they're going to be able to live. Refrigeration is approximately about 4 degrees Celsius. That slows spoilage by limiting the growth of your fast-growing mesophiles or psychrophiles. Psychrophiles can grow. Psychrotrophs can grow, but oftentimes they'll grow slower than they would otherwise. We can use freezing to preserve food. Freezing doesn't typically kill microbes. It can kill microbes, especially if ice crystals form, but it can stop the microbes from growing. And then the temperature also is going to influence different microbes in causing disease. So there are certain diseases that microbes can only grow at the cooler portions of the body. So an example is Mycobacterium leprae grows in cooler regions. So if it's going to cause leprosy, it's going to affect the extremities first. So your hands, fingers, feet, ears, toes are going to be the things that are going to be most affected by leprosy. The next environmental requirement is oxygen or atmosphere. So you need, so many organisms need oxygen to grow, but there are many more that don't utilize oxygen at all. So when we are growing microbes, we can actually determine if the microbe is an aerobe, an obligate aerobe, or if it is not an aerobe, if it's an obligate anaerobe, just by producing or taking certain types of media. When we autoclave the media, we get rid of all the oxygen. And as it cools, the oxygen is going to start settling into the media at the top. So at the very bottom, there's going to be no oxygen. And so organisms that are growing are going to grow at the top if they're aerobic, and they'll grow at the bottom if they're anaerobic. So the position where the organism grows is going to determine how much oxygen it needs. There are 5 different types of organisms associated with oxygen requirements. We have obligate aerobes, which require oxygen, cannot survive without it. Facultative anaerobes, which utilize oxygen. They do not require it, but they tend to grow better with it. Obligate anaerobes cannot use oxygen, will not survive in the presence of oxygen. Microaerophiles require small amounts of oxygen, about 15% of the oxygen in the environment. Or I'm sorry, about 85% of the oxygen in the environment. So they grow at lower oxygen levels, about 15% lower than your obligate aerobes. And then you have aerotolerant anaerobes, which are able to grow in the presence of oxygen, but they do not utilize oxygen. And so here is a medium. So this would be a broth medium. The one that we typically use is called fluid thioglycolate. And at the very top here we have growth, which means that these are obligate aerobes. Facultative anaerobes grow best at the top where oxygen is, but they can grow throughout the whole thing. Obligate anaerobes grow in the absence of oxygen, so down here. Microaerophiles need about 85% of the oxygen that your obligate aerobes need. So they grow just below the top. And then aerotolerant anaerobes can grow throughout the entire media very effectively. The difference between these two is you can see a lot more growth at the top where facultative anaerobes like to use oxygen. Aerotolerant anaerobes don't care if oxygen is there or not. So how can they do this? How can we have these different types of organisms? Oxygen, in a nutshell, is very toxic, but animals and many other organisms utilize oxygen, and we require it. But because it is so toxic, we have special enzymes that protect us. And so 2 of those enzymes are superoxide dismutase and catalase. So obligate aerobes have both. Facultative anaerobes have both. Obligate anaerobes have neither. Microaerophiles have both. Aerotolerant anaerobes have superoxide dismutase. So when we talk about oxygen, think about the fact that all animals require oxygen, but not all organisms require oxygen. And oxygen is not one of the gases that was in the environment in our initial or our original atmosphere on Earth. So oxygen is actually a very toxic chemical or toxic atom. Oxygen gas is not toxic, but when it goes through aerobic respiration, it can become a dangerous free radical that can then attach to the plasma membrane, attach to cell walls, attach to the DNA, and cause all kinds of harm to the cell. Hydrogen peroxide is often produced as a byproduct of getting rid of those free radicals. And hydrogen peroxide also can do damage to our cells. And so we have to have enzymes that can get rid of these superoxides and hydrogen peroxide. Superoxide dismutase inactivates the free radical superoxide by converting it back to oxygen gas, which is not toxic to us, and producing hydrogen peroxide. So hydrogen peroxide then has to be converted also because it can damage our cells. So it is converted to oxygen gas and water, which is not damaging. I'm going to stop here and we'll get into pH in the next video. Bye.